Welcome to Mr. Chalk's Revision Tips. In this video we will look at water potential, osmosis and some experiments that look at osmosis. So osmosis is a term that describes the movement of water through a semi-permeable membrane from an area of high concentration to an area of lower concentration. Some key things that we need to think about and look at in this video are water potential. So this is talking about the concentration of water. Osmosis in cells and the effect that osmosis has on cells. And more importantly, how osmosis affects plant cells. So, like we've just previously said, osmosis is a term describing the movement of water from an area of high concentration to an area of low concentration through a semi-permeable membrane. The water moves towards a high concentration of solute with the effect of equalizing the solute concentration across the permeable membrane. From the point of view of the water, it undergoes diffusion, moving from a high concentration to a low concentration of itself. So osmosis is also a form of passive transport, as it does not require any energy to take place. Water potential is the potential energy of water per unit volume relative to pure water in reference to its conditions. Water potential quantifies the tendency of the water to move from one area to another due to osmosis, gravity, mechanical pressure and the matrix effects such as in capillary action. So water potential measures the concentration of free water molecules. It is a measure of the tendency of these molecules to diffuse to another area. The more free water molecules, the higher the water potential. Water diffuses by osmosis from a region of high water potential to a region of low water potential through a water potential gradient. Osmosis can therefore be defined as the diffusion of water from a region of high water potential to a region of low water potential through a partially permeable membrane. So, when a cell is submerged in water, the water molecules pass through the cell membrane into the cell. So, if the cell is placed in a solution of low water potential, the water will diffuse out. This causes the cytoplasm to shrink and the cell to become flaccid. If enough water leaves, the cytoplasm will pull away from the cell wall and the cell will become plasmidized. So this is when we're thinking about plant cells. Since cells contain various biological molecules, such as sugars and salts, they have a water potential lower than 0 kilopascals. Water may move in or out of a cell depending on the water potential gradient between the inside and the outside. So when water diffuses into a plant cell, when it's placed into a solution of higher water potential than inside of it, the cell contents will expand. However, since plant cells are surrounded by a strong cell wall, they will not burst. The cell contents will push against the cell wall and the cell will become turgid. So we can see from that diagram there that in a hypertonic solution, water will move out of the cell and the cell will become plasmidized. In an isotonic solution, so that's a solution where you have an equal water concentration inside and outside, water will just move in and out and the cell won't get any bigger or smaller. And in a hypertonic solution, So where the water potential outside of the cell is a lot higher than inside, water will move into the cell and cause it to become turgid. So lots of experiments can be done with osmosis. Quite often we'll use something like a potato chip. So in this example, scientists investigate the effects of osmosis on liver cells. So they either observe the cells or tissues placed in solutions of different concentration. They measure the changes in the cylinders or discs of this potato or beetroot. The cylinders will have larger mass than discs, so scientists will have a larger measurement to work with. In this experiment, it investigates the effect of different concentrations of sucrose on potato tissue. It could also be carried out using salt sodium chloride solution instead of sucrose. So we can see from this graph that in terms of the mass change, the lower the sucrose concentration, the higher the water potential will be, so therefore the water will move into the cell. And conversely, the lower the water potential, so the higher the sucrose concentration, the more water will move out of the cell and therefore cause its mass to go down. So in a graph like this, we can go and work out what the water potential inside of the cell is, because it's where the lines all intersect. So we can see from this graph that the potato cells must have a water potential of 0.3 or a sucrose concentration of 0.3 moles per decimeter cubed. Thanks for watching.